Next, I will be talking about secondary envelopment. Secondary envelopment is the final stage before the virus is released. What happens is the viral capsid leaves its envelope in the outer nuclear membrane, and when it enters the cytoplasm, it will get a new envelope by budding into the membranes of the Golgi. So going back to figure 1, if we look here at the herpes virus egress, right here in this section is where secondary envelopment is occurring. We see the viral capsid budding into the Golgi and then exiting with its new membrane. Figure 2 is just an electron micrograph of secondary envelopment. So if we're looking at the first picture here, you can see these dark dots are the viral capsids, and these C shapes surrounding them are the trans-Golgi network membranes. And so you can see that this is the process of budding here, and in this second photo, you can see that the capsids are fully enclosed in the membranes now. So for herpes viruses, the main membranes involved are the cis-Golgi, the medial Golgi, trans-Golgi network, and endosome membranes. And these membranes become dramatically rearranged during the process of secondary envelopment. So now looking at figure 5 from the review paper, this is a more in-depth diagram of secondary envelopment. And so you can see these fork, spoon, and knife looking shapes. These are glycoproteins in the trans-Golgi network membrane. And over here, as you can see in the key here, we have the tegument proteins from the viral capsid. And so both of these proteins, the glycoproteins and the tegument proteins, interact with each other in order to induce the budding into the membrane. So looking specifically at the case of human cytomegalovirus, we see that the interaction between the glycoproteins and the tegument proteins causes the membranes to rearrange and to form something called assembly compartments or assembly complexes. And what these do is they help form the virions, and they also are a major part of the virus egress. So I wanted to point out human cytomegalovirus before, or HCVM, because there was actually a study done on the relation between the HCVM assembly complex and the egress of herpes virus. This study was done by researchers in 2007 at the Lerner Research Institute at the Cleveland Clinic Foundation. Their main goals were to study the relationship between the assembly complex and secretory organelles, also to see how time played into the remodeling of the cytoplasm after HCVM infection, and then also to see if they could figure out the 3D structure of the assembly complex. And then lastly, these would provide implications for HCVM and its egress. Human lung fibroblast cells, or HLF cells, were used in this study since herpes virus can occur in fibroblasts and use them as host cells. The main techniques used in this study were confocal immunofluorescence and immunoblots. Immunofluorescence was also used in the study on de-envelopment. So again, with immunofluorescence, we're looking at staining particular molecules with a fluorescent dye in order to track them for study. With immunoblots, what we then do is use these tracking antibodies specifically on a membrane, and gel electrophoresis is also used with immunoblots. So specifically here, I decided to look at figure 3 because it's a good example of immunofluorescence. And so we're looking at 3 different types of stains that they used here. They used a blue DAPI stain to track DNA, and they used a red MAB stain to look at the ER, the Golgi, the trans-Golgi network, and early endosomes. And then a green Revit polyclonal antibody was used as a Golgi marker. So here in these photos, we can see that in this first column here, they were looking at the location of different organelles and also then the DNA. And then in this second one, they were looking at the Golgi markers to see where the Golgi was and also the DNA. And then what they did was that they merged these two photos together to see where the lineup occurred between the position of the organelles and also the Golgi. This bottom photo here just gives us some perspective as to the patterns that we can see with these different organelles and where they are. What I found really interesting in this paper was the 3D structure of the HCVM assembly complex, and that was delineated in figures 5 and 6 in the paper. And so on the right here, we have the original confocal microscopic images with the immunofluorescent staining. And then on the left here, we have the reconstructed images. And so these bottom photos here are cross-sections of the top image, and you can see where they cut with this dotted line here. And the really interesting thing about these photos is that we can see how the organelles are nested in each other here. We have the DNA surrounding the Golgi, which then surrounds these early endosomes. Then in figure 6, what we have is just a drawn diagram of the photos that they got from figure 5. Now this all relates back to secondary envelopment because once we know the structure of the assembly complex and this interesting nesting of the organelles, we can see that these HCVM viral capsids are actually budding off, and then we can see the order in which they're going to encounter the various organelles and the Golgi and trans-Golgi network, and how they might become re-enveloped before being released and continuing on in their egress. So overall, after looking at our review and our individual papers on primary envelopment, de-envelopment, and secondary envelopment, some of the major points that we wanted to take away were that the herpes virus egress is really dependent on interactions and reorganizations of cellular membranes, and then also that the proteins involved in this are really huge factors in the alteration of the nuclear lamina and also membrane fusion, which are central to the egress of the herpes virus. And this relates really well back to the paper that we read on Ebola and just the importance that the cellular membrane plays in transferring the RNA from the Ebola to the host cell. And just like that, we can see here the role that the membranes are playing in the transfer of the herpes virus to its host cells. In addition, we have both of these studies using immunofluorescence as one of their main techniques for tracking the various components of the virus and the cellular membranes. And finally, in regards to herpes virus egress, some areas that could be further researched are the specific roles that the BIOL34 and 31 proteins and viral glycoproteins play in primary envelopment, and then also to further study the tegument layer and tegument proteins in secondary envelopment, and to really specify their interactions with various domains of glycoproteins.